the announcement of the most detailed 3D high-resolution images of the human fetal brain. We are truly delighted to share this remarkable milestone with you. To begin, I invite Professor Monashankar Shivaprakasham, Head of the Sudha Gopalakrishnan Brain Center at IIT Madras.
Welcome, everyone. Since we are sort of assembled here for something which is quite momentous, we thought we should give you some background about the center itself. So we have a very brief presentation. Brain Center was started in 2022, March, and it got seeded by support from our beloved alumnus, Mr. Chris Gopalakrishnan, and the Office of Principal Scientific Advisor, Government of India. And this is a very unique center in the sense it combines neuroscience, technology, engineering, medicine. And as a result, this center is today working on the cutting edge brain sciences. And as you can see, the center has, after the seed support from Chris and the PSA office, it has attracted support from various philanthropists and industry, and the list is there. And what we are doing is we are imaging whole human brains at cellular resolution. That is what we are doing. And we are doing it at a certain scale and speed, which is perhaps the fastest and the largest in the world. So we are processing almost one large brain per month. That is the pipeline and throughput with which we're doing. Some of you who have not seen the center, you're welcome to visit the center anytime. So we start with a brain. We slice this brain at very thin sections, 10 to 20 micron. That's about 1/10 to 1/5 of the human hair. And then we do a series of processes using a lot of sophisticated robotics, instrumentation. And then we microscopically image it at about 1 to half a micron, which is 1/100th of a human hair. And now you are entering the digital domain. This brain has now been digitized. Now you have to put it back together into a 3D. Right? Essentially, we are imaging the entire brain at 3D at cellular resolution. And we have been successful in that. And all this happens because we have developed an indigenous technology platform. இது முழுமையா இந்தியாலேயே ஐஐடி மெட்ராஸ்லேயே சென்னையிலேயே தயாரிக்கப்பட்ட தொழில்நுட்பம். And it starts literally from how do you freeze the brain? Because when you have a brain which is extracted post-mortem within 6 to 8 hours, how do you freeze it? How do you cut it? All these instruments are completely developed by IIT Madras indigenously. And as a result, this is now one of the most sophisticated platforms in the world in imaging whole human brain. So this is to give you a sense of the process. This is how brains are, which we extract, both normal brains as well as brains with specific known diseases. Right? Diseases right from fetus, infections, developmental disorder, stroke, dementia, Alzheimer's, various brains. And today we have close to about 220, 230 brains from various sources, from medical collaborators, which I'll talk about in a while. When we section them and we image them, this is the kind of serial sections you see. And this is the level of detail. When we say cell resolution, what you're seeing there is a zoom in version of a cell of the entire section, which you will see there. Now comes the real challenge, and today it is one of the biggest achievements, is now we have all this, we are able to create a 3D view of the entire brain. What you're seeing is actually the world's first 3D human fetal brain at cellular resolution. And now the announcement today is not just one brain, it is 5 brains, which is this, what we call the second trimester, 14 weeks to 24 weeks fetus. I want to really stress this fact that all this was possible because we have a very truly a large team of multiple disciplines, medical collaborators, Mediscan. DHARANI is the name of this thing. So today, this is the largest publicly available digital human fetal brain in the world. We have published it in Journal of Comparative Neurology. It is 133 years old. The editor will correct me if I'm wrong by even 1 year. 131, 1891, so 133. 133-year-old journal, which is continuously published. And we picked that also to show that what is the significance of this. This is a giant leap in terms of the quality of the data and the availability of the data. It is actually accessible to anybody, not just researchers or our collaborators. It is accessible to right from the top brain researchers to high school students today. Right? And we have also made it in a way that you will be able to see the cell level resolution and also the 3D level. All these links have been made public to anybody who can access it, and it's also being shared with media. In addition to the cell resolution, we have created the maps of the human brains. So this is a really hard, a very, very laborious and also technically challenging process, because you have a brain with hundreds of billions of cells, and we have created maps for the first time, which has brought out 500 plus regions across 5,000 odd sections of fetal brain. Okay? And you will see a glimpse of it here. If somebody can play it, just somebody has to click on. So what you see on the left-hand side is the cell resolution images. On the right-hand side is the maps that we have created. The maps of the fetal brain for the first time across the entire second trimester at cell level resolution. So this is a very significant milestone for IIT-Madras, for neuroscience in the country. And you'll hear from the editor very soon and why this is significant at a global level neuroscience as well. So thank you very much. We will get questions after the entire presentation is over. Thank you.
Dr. Susanna Herculano-Hausel, Editor-in-Chief of the Journal of Comparative Neurology, to share a global perspective on the significance of this research.
I'm Susanna Herculano-Hausel. Today I have the pride and honor to announce that the Journal of Comparative Neurology, of which I am Editor-in-Chief, one of the, it's the oldest continuously published basic neuroscience journal, is the journal bringing to the scientific community a monumental contribution that comes not from the usual Western power players in the business of generating knowledge about the brain, but from India. Today, the Sudha Gopalakrishnan Brain Center of the Indian Institute of Technology, Madras, is gifting humankind with DHARANI, a freely accessible digital micrometric resolution collection of three-dimensional atlases of the developing fetal human brain. DHARANI, named in tribute to the Hindu goddess of the earth and of beginnings, stands for a three-dimensional developing human brain atlas resource to advance neuroscience internationally. It's an acronym. DHARANI is the kind of tool that opens new horizons, which is exactly what maps do for navigators. A brain atlas is a stack of maps of the brain, and each map of the brain is a chart that represents side by side on a flat surface of paper or screen what exists side by side in a flat section of the brain. Because the brain, unlike the surface of the planet, is a complex three-dimensional structure, the intricacies of its anatomical organization can only be captured by cutting it into series of very thin sections that can then be colored for contrast, placed into a system of coordinates, described, annotated, and then finally organized systematically into a stack that reconstructs the third dimension of the brain. The team of dedicated scientists, engineers, and computational technologists who created DHARANI did that for 5 developing human brains at 5 different ages during the second trimester of gestation, when the brain is rapidly growing in size and quickly increasing in complexity. With its thousands of plates that capture with superb quality the growing intricacies of the developing human brain, DHARANI will give neuroscientists around the globe will guide neuroscientists around the globe in their endeavors to understand how the human brain comes to be what it is and to do what it does. This monumental contribution is all the more remarkable because it was entirely built at the IIT-Madras during the COVID pandemic and was created with less than 1/10 of the funds that powered its US counterpart, the Human Brain Reference Atlas of the Allen Institute for Brain Science, which was also published in the Journal of Comparative Neurology in 2016. DHARANI is now the largest publicly available digital data set of the fetal human brain. And as I speak, the Sudha Gopalakrishnan Brain Center is welcoming experts from the United States, Romania, South Africa, and Spain to explore DHARANI together and begin to build international scientific relationships of the kind that bring humankind together in friendship and collaboration in the common quest for self-knowledge. This is the human brain at its best, collaborating to build together what no single human brain could possibly achieve alone. I look forward to seeing what the Brain Center, the IIT-Madras in India, will bring next to the table of human brain cartography as DHARANI continues to grow as a platform that allows us to visualize in 3 dimensions how from simple beginnings, this most complex of structures is formed, that is the human brain. Thank you.
Dr. J. Kumata, Dean of Neonatology at Savitha Medical College and expert advisor to the National Health Mission, Government of Tamil Nadu, to speak about the impact of this work on Indian healthcare.
Good morning to all the press and the IIT professors here. And Kalai Vanakkangal. This research of Sudha Gopalakrishnan's Brain Center is very, very path-breaking research for clinicians like that. See, we are basically pediatricians, neonatologists, or adult physicians. We are very, very intrigued about so many diseases which affect the brain. This affection of the brain or the disease of the brain starts from the womb. That means even when the baby is in the mother's uterus, it starts problem in developing. And also we want to know how the normal brain develops. Because with advanced technology where they have done the sections to the tune of 20 microns and studied at less than 1 micron, that gives more insight to our clinician that how the developing brain behaves at various gestation and why certain babies when they are born do have certain problems and why some babies do not have. So this also they have shown that there are many new findings which was not in the previous atlas mapping during the process of development. That means our babies, whomsoever the brain has been studied, showing a new changes and new development which is occurring little earlier than what we expected. That means if we expected a particular development of the brain at 14 weeks, they are seeing it little earlier or at 17 weeks, whatever. So this gives a lot of insight. And coming to our health of India, see, our Prime Minister also is promoting original research in our country. And during COVID period, we proved it. And in fact, this study was started during the COVID period, despite all the calamities. And we pulled together. I really appreciate the IIT team with other collaborators who have done tireless work on this. And again, see, the major areas it would help is the developmental disorders we see in children. Some babies go for cerebral palsy and permanent damage due to some hypoxic change. But some babies, despite having lot of hypoxia, behave normally. So we don't know why a particular baby goes for cerebral palsy. Same way, certain babies go for certain developmental disorders. They are born all right. So autism is one of the important things where we have not found why this baby was born looking normally for the first 9 months of life and goes into autistic behavior later on. So that is another area where we could help. And again, coming to the mental health disorders, see, disorders like depression or bipolar disorders or even schizophrenia, which comes in a particular families. What are the genetic disposition? In fact, studies have shown certain brains, the difference between a primate brain and our brain, the cells are almost the same. But how the gene differentially gets activated in the brain is what makes a human reaction or human behavior different. So all this, they have done it at a cellular level now. When they go for the functional level next in the chemical processing, they might be able to give us an answer. And again, what should be appreciated is the spectrum of brains they are studying, right from in utero brains, stillbirth brains, to neonatal deaths also we have taken, and they are planning to move to pediatric age and certain adult brains also. And adult normal brains who would have died due to accident, and adult who had diseases, particular disease. So we will know how the brain, what are the brain changes in specific diseases. So these are things I think which would help.